Hello, and welcome to part one of the lecture on chapter two on chemistry. It's microbial chemistry, but it also is the chemistry of pretty much all life forms. There are some large organic molecules that all living organisms use, and there's four main categories, and that's what we're going to go over in this lecture. So organic, what do we mean by that? Where the term came from. Originally, when people used the term organic chemistry, what they were talking about were the molecules of living organisms. That's why it's called that. It has come to mean molecules that contain carbon and hydrogen. That's organic chemistry. But where it came from originally was these are the compounds that are made by organisms. We are going to concern ourselves with four main categories of organic macromolecules, macro meaning big molecules. There are four main categories. I know, have heard of three of these at least because we talk about them all the time when we talk about our diets. Like these are the so-called macros that people keep track of. I have numbered them here just to be able to keep track of where we are in the lecture. So the first category, there it is. The first category we're going to cover are the lipids. These includes the fats and oils. And well, we'll go into detail there. But when you talk about diet, that's what people refer to as that macro is the fats. Okay. Then the second macromolecule we'll deal with are the carbohydrates, sometimes referred to as carbs. And then the third category are proteins. And finally, the fourth category are nucleic acids, DNA and RNA. So I said there were three macros that people keep track of, and it's the first three: lipids or fats, carbs, and proteins. That's what people generally count. And the reason that we don't count nucleic acids, it's not that they're not important, they are super important, but here's the thing. If the food that people are eating has ever been alive, then it's got nucleic acids in it. So yeah, you're getting some nucleic acids in when you eat plants or animal bits. And so yeah, they're taken care of. We just don't generally count the amount of grams of nucleic acids that we're getting in a day. Okay, the other term I want you to know related to this is three of these: carbs, proteins, and nucleic acids. All three of them are made of repeating structures. So carbs have a particular kind of structure that repeats. Proteins have a particular kind of structure, the amino acids that repeat. Nucleic acids have a particular structure, the nucleotides. I know you know these terms, you've heard them before. What we call all of those together are monomers. Mono meaning single, like the individual single units, the basic building blocks of these. So carbs, proteins, and nucleic acids follow that pattern. Lipids are a little bit different. There's something else that the lipids have in common that make up kind of hang together as a group, where carbs are all made of the monomers called sugars, proteins are all made of the monomers called amino acids, nucleic acids are all made of the monomers called nucleotides. Lipids are different than that. So let's have a look at lipids first. So we'll start with number one. As I just mentioned, they're not composed of regular subunits. There's a number of different types, and I'll show them to you. What they all have in common is that they do not play well with water. They do not interact well with water. I mean, just think, the last time you made salad dressing or had salad dressing, you had to shake it up, and the oil and the vinegar, they don't mix. Oil and vinegar is largely water. They separate from each other. So what these lipids all have in common is that they are hydrophobic, do not interact well with water. And then within the lipid category, we have a number of different kinds of lipids that we're interested in. What we refer to as fats, like nutritional fats or storage fats on our body, these are largely made of what are known as triglycerides. And our cell membranes are largely made of phospholipids. The triglycerides and phospholipids are they're pretty similar in structure. The triglycerides, as you might guess, have 3 fatty acid tails. There's where the tri comes from. And then the phospholipids, they just have the 2 fatty acid tails. And we'll talk about those fatty acid tails. And then waxes. And finally, we'll finish off with the steroids. All right, so triglycerides. These are represented in stick drawings like this. Whenever you see a squiggly line like this, anywhere there's a corner or there's a change in angle, there is a carbon atom. And so here, this representation actually shows the carbon atoms, but this is the kind of shorthand for that. Let's see, what can I tell you about that? So anywhere there's a carbon, remember that carbon is like, it's like your basic Lego when you're, building a complex thing out of Legos. It's the basic one. It's a basic building block. You have lots and lots of carbon. And then carbons can make 4 bonds. So here, this carbon is going to be bonded to the 2 carbons on either side, but it's also going to be bonded to a hydrogen, 2 other hydrogens, one above and one below. Why am I telling you that? Because of this. Do you see how there's a kind of wiggly line and a zigzag? As long as it's consistent zigzag, what that means is that there are single bonds between the carbons, and this thing is saturated in hydrogens. It's holding on to as many hydrogens as it can possibly hold. And so when we see this relatively straight zigzag, we say that is saturated. It's a saturated fatty acid. And it's not that hydrogen is bad. It just means that there's lots of the hydrogens on there. And the way you do that is by only having single bonds between the carbons. That's how you get this straight thing. Now I want you to think for a little bit about some stuff in your kitchen. There's probably some saturated fats and some unsaturated fats. And the way you can tell the difference at room temperature is largely what form they take. In other words, if you have coconut oil or bacon grease or some other solid fat, butter, these are largely saturated fats. And at room temperature, they're kind of solid, semi-solid. In contrast, you may have some things in your pantry, some oils. These are also lipids, triglycerides, but they at room temperature are more liquid. And let me see if I can try to explain that quickly. This is the trick to unsaturated fatty acids. There's a double bond somewhere, at least one double bond, and that puts a kink in this chain. So the carbons can no longer each hold on to 2 hydrogens. These 2 carbons, they're double bonded to each other, so they can each only hold on to 1 other hydrogen. So that's unsaturated. It's not holding as many hydrogens as it could possibly hold. That double bond puts a kink. So instead of the carbons, holding on to each other side to side, they've got 2 of the bonds going in one direction. And so it puts a little kink in the chain. And so what I want you to think about is if you've ever had a stack of books or a stack of boxes that you can put on top of each other, that's a pretty stable thing. But if you've got something that isn't quite straight, it used to be we'd have a 3-ring binder, but I'm trying to think of what might put a kink into your pile of papers or something, like a spiral bound notebook or something that makes things not lay quite flat. You know that if you keep stacking things on top of this kinked structure, they're going to slide off. That is what I want you to picture when you think of oils or unsaturated fats. They're less stable at room temperature. They fall apart more easily. So that's how you can just visually assess whether something is a saturated fat or an unsaturated fat at room temperature. The saturated fats stack on top of each other really easily. And the unsaturated fats, they have a kink in them, so they don't stack as well. And so those are the things that tend to be liquid at room temperature. Now, obviously, that changes if you change the temperature. You can get butter to melt, for example, if you heat it up, and you're not suddenly making it, saturated into unsaturated. That's not the way that works. But that room temperature structure is just a short way of quickly assessing what kind of bonds you have in this thing. Okay, so those are triglycerides. So I mentioned the 3 fatty acid tails, and the glycerol part is this part. It's a 3-carbon structure, and each one holds onto one of the fatty acid tails. In case I mention it later, the bond between the glycerol and the fatty acid tail is referred to as an ester bond. There's an oxygen in the middle, for those of you who love chemistry. Okay. And oh, so this this one shows the structure of some fatty acids, some saturated fatty acid tails. And it's just a number of carbons repeating. And both of these are saturated. I'm not going to go into detail, but the different lengths of chains have different names. Now, this is what I want to show you. This is an unsaturated fatty acid, mono unsaturated. That means there's one place where there's a double bond. So I've got this straight, relatively straight chain. Then there's a double bond that puts this big kink in this chain, and then it continues. And let's see, if there's more than one double bond, we call this a polyunsaturated fatty acid. So here's a couple of double bonds. So again, these would tend to be liquid at room temperature. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9. So you've probably heard of omega-3 and omega-6 fatty acids. Probably omega-3s are the ones you've heard of the most. Where that number comes from, these carbons are numbered. This one is the alpha carbon, the one that's closest to joining the structure, and then there's a tail end, that's the omega carbon. And so you count from the omega carbon where this double bond occurs, tells you what kind of fatty acid this is. So this is an omega-9 fatty acid, unsaturated, monounsaturated fatty acid. And this would be 1, 2, 3, 4, 5, 6. This is an omega-6 fatty, polyunsaturated fatty acid. Now, remember I mentioned the phospholipids. These are really important in the cell membrane. We've talked about them before. They have 2 fatty acid chains instead of 3, but they still have this glycerol molecule. So here's fatty acid chain number 1, here's fatty acid chain number 2, and then instead of having a 3rd fatty acid chain, instead there's this phosphate group. This head, this phosphate and 4 oxygens here, this thing is polar, hydrophilic. So this is the part that interacts with water really well, while the tails are hydrophobic. So I'll often describe this as the lipid tails turn in towards each other, trying to get away from the water, and then the heads will point out into the water outside of the cell and into the water inside the cell. What I'll often refer to as the Oreo cookie layer of the cell membrane. Same thing goes for these tails. If they are saturated, that means they're no double bonds, pretty rigid, and they're pretty stable. And double bond means that there's going to be some more flexibility. Fascinatingly, how many of the tails are saturated and how many are unsaturated is often related to what temperature that organism evolved to optimize conditions. So in colder climates, you'll find organisms have more unsaturated fatty acid tails. And in Warmer climates, you'll find they have more of the saturated. So think about this. So saturated, warmer temperatures, think coconut oil, more solid at room temperature. In contrast, if you're thinking about fats that are more unsaturated, for example, oily fish, like salmon, the salmon is kind of a cold water fish. And so it's the fatty acid tails that you find in salmon tend to be more unsaturated fats. Amazing. All right, we'll continue with lipids. This category waxes. There's the thing they have in common with the other lipids is that they're hydrophobic. They don't interact well with water. It's just one long chain fatty acid linked to an alcohol. Anyway, they don't have a hydrophilic head like the phospholipids do. So they're completely insoluble in water. That makes waxes really great for protecting microbes from drying out, from desiccation. And finally, the last category of lipids, man, they don't look like the others. They don't have fatty acid tails. What these all have in common is this general four-ring structure, largely made of carbons and hydrogens. That's the definition of these organic molecules. And the one that's the most common in our body is the cholesterol, cholesterol. So sterol is this four-ring structures, what they all have in common. We find it here in the creamy part of the Oreo cookie layer of the cell membranes. So all of our cells have cholesterol in the membrane. It's a really important part of cell membranes. But they have other functions as well. And I'm not going to have you memorize all of these pathways, but a few things that I want to point out. Cholesterol is kind of the basic building block for making progesterone, for making testosterone, for making estrogen, and aldosterone. It's one of the important hormones that's used to regulate water balance in our bodies. But you have got to have kind of this basic cholesterol to work off of. So those are the so-called steroid hormones. And I think that is a good stopping place for the end of part 1. See you over in part 2. We'll continue with carbohydrates.